The technical aspects of methylation that have to do with where methylation is in the genome. We talked about the maintenance of methylation, which is more than a technical aspect. The fact that methylation inhibits gene expression. And today what I'd like to do is to get into the more biological aspect. And I think the interesting thing that we should all be doing, actually, is looking at the biological picture. And in the case of methylation, even though a lot was known about methylation in the 70s and 80s, we didn't realize it then, but we were really far from understanding what the role of methylation is in vivo. And I dare say that even today, we don't really understand completely the role of DNA methylation in gene regulation. And for sure, there are people who often say that DNA methylation doesn't contribute anything to gene regulation. So we're going to talk about this a little bit today. As it turns out, I'm going to have to try and convince you that DNA methylation plays a role. But I'm trying to build up a little army of believers, so maybe you'll be able to help us out with that. Okay, so the first thing I want to tell you is something about how methylation works. We talked about the fact that DNA methylation inhibits gene expression. It was shown in tissue culture. In a little while, I'm going to show you how, after many years, we succeeded also in demonstrating that DNA methylation inhibits gene activity in vivo during normal development. We'll get to that. The question is, how does DNA methylation work? What makes it inhibit gene expression? And I think that the early people who worked on this, including our own laboratories, were really of the opinion that DNA methylation sort of works like a mutation. DNA methylation of C looks a lot like T, the nucleotide thymidine. And the concept was that if you change a C to 5-methyl-C, it now becomes more like a T. And then all the proteins that have to recognize the DNA, and they recognize the DNA on the basis of sequence, and they see this 5-methyl-C instead of C, they won't be able to bind, or they won't bind correctly. And that this is the way DNA methylation affects gene expression. As it turns out, this is probably not the case. There are a few sites in the genome, a few transcription factors, a few factors that bind to DNA which are inhibited by the presence of 5 of the methyl group on the cytosine or cytosines that are located in their site of recognition. But this is the exception. Basically, it turns out methylation works by affecting chromatin structure. Okay? And one of the first experiments in this direction was done by Ilana Keshet. And it was a very, very, very simple experiment based on the type of experiments that I showed you last time. And that is, what we did was we took DNA. In vitro, we methylated the DNA, or we didn't methylate the DNA. We inserted it into cells. We isolated clones that have this insert, either methylated or unmethylated, and then asked whether these sites, these DNAs that have integrated into the genome, are now accessible or not accessible, using DNase sensitivity in nuclei as an assay for accessibility. Okay. Now, I must give you a little introduction to the technique. It's important to understand the technique here. Okay? So the technique for putting DNA into cells, transfection, or what's often called DNA-mediated gene transfer, is done by taking DNA and adding it to cells in the presence of some sort of vector that provides selection. So in the experiments I showed you before, the vector for selection was often the TK gene, and we introduced it into TK- cells. Over the years, we saw the development of many other selective markers, in particular all sorts of antibiotics, neomycin, puromycin, hydromycin. Today there are even many more. This allows one to basically select the cells that are capable of taking up DNA. That's the way the selection works. Cells in culture, for some reason that has to do perhaps with the cell cycle, at certain stages or at certain parts of the tissue culture plate, they're capable of taking up DNA. And by including together with your DNA a selective marker, you basically select for those cells that are capable of taking up DNA, and the DNA goes into the cell. Usually during transfection, all of the DNA integrates into one site in the genome. Okay? Now, in order for the DNA, in order for the experiment to work, the DNA has to integrate into a site that's accessible, that's open, because otherwise the selective marker won't work. So what happens is when the DNA gets into the cell, it recombines with each other. All the molecules sort of recombine. It generates a very, very large molecule, and it's that large molecule that integrates into the DNA. In order for the DNA, for us to detect these cells that integrated DNA, the selective marker has to be in an open conformation so that it can be expressed. Okay? So in general, if you just take DNA and transfect it into cells and then go and test whether that DNA is accessible, and even the DNA that's associated with it, it's going to be accessible because you're selecting for cells like that. Okay? And indeed, that's what happens. So if you take, these are all sorts of pieces of DNA that were co-transfected with a selectable marker. The names of the DNAs are not here, but they're mostly DNAs that have nothing to do with animal cells. They're taken from bacterial cells, they're taken from viruses, from phages, things that have nothing to do with animal cells. So here's an example DNA that's unmethylated, and we did a DNase analysis. That means taking nuclei, digesting them with DNase at various concentrations, increasing concentrations, then doing a southern blot to detect the DNA. And as you can see, as you increase the concentration in the nucleus of DNase, the DNA gets digested. Okay? So this is sensitive. You have to look at it with a marker that you know is sensitive. And you see that this DNA is sensitive. I think this is M13 DNA. It's a bacterial phage. When that DNA was methylated, it was insensitive to DNase. Right? So as expected, this is sensitive. And every example that you put into cells, it's sensitive. It has to be because you're selecting for that. Yet if the co-transfected DNA was methylated, it was not sensitive to DNA. So it was true for this DNA and the next DNA. They're all non-animal cell DNA, completely resistant to DNase digestion, even though the unmethylated copy is sensitive. So this experiment showed that if there is DNA methylated in the cell, okay, and here again, transfection is a wonderful system because you control in vitro what's going to be. The only difference between putting in this DNA or this DNA is that this one's methylated. It's the only difference. It goes in, and because it's methylated, it's resistant to DNase. And that suggested that the DNA within the cell is the same way, that if the DNA within the cell is unmethylated, it'll be open, and if the DNA within the cell is methylated, it'll be closed. And we'll talk about this a little bit later, but it's clear that DNA methylation affects gene structure. We don't know a lot about what this means in terms of DNA. What does DNA sensitivity mean? But it has something to do with accessibility. A lot of things go into this. It's a complex process of building chromatin, most of which we know very little about. But it's clear that methylation affects this. Another way of looking at this, and this is an experiment that was done by Sharon Aden, and Tamar Shimshoni, in which we looked at histone acetylation. So in this experiment, you take nuclei, you make chromatin. We treated nuclei with micrococcal nuclease to make nucleosome-type particles. You take these nuclei, and you can see that you really get mononucleosomes. This is now fractionated on a sucrose gradient. You get mononucleosomes, dinucleosomes, trinucleosomes. We take either the mono or the dinucleosomes, and we precipitate them with an antibody against acetylated histone. Okay? And usually, active regions are acetylated. Inactive regions are generally unacetylated. So what we did was, we took, these are controls. So we took a gene that we know is repressed, and we look at the input DNA at 2 concentrations, and then we look at the bound DNA with 2 concentrations. Okay? And you can see that this is relatively not enriched, the bound DNA, as compared to the input DNA. Okay? After precipitating with an antibody against acetylated DNA. On the other hand, if we look at an active gene, here's the input, which here is relatively low, and the bound is relatively high, because active DNA, like beta-actin, is bound by acetylated histones. So then we took this system, we took nucleosomes, we performed the controls, we did the chip analysis, isolated the bound and input DNA, and then analyzed DNA that had been inserted into these cells by transfection, either unmethylated or methylated. The same approach. Okay? And without going into a great deal of detail, you can see somewhere over here, this is the inserted DNA. We put in TK DNA. So you can see that when it's unmethylated, input bound, it's in an active conformation. Again, for the same reason, because when you transfect into cells, you basically select for activity. So it's input versus bound. You can see the bound is more than the input. Yet when it's methylated, input and bound look the same here. It's not enriched. So the methylation is affecting histone acetylation. Right? Turns out that this also affects the, I'm not going to go into this, but it also affects the DNA sensitivity of the gene. Okay. So the picture is that the way DNA methylation works is that DNA that's unmethylated, following replication, okay, following replication, where the DNA has to be repackaged, it's repackaged in a relatively open conformation. I don't know if you can see it, but this is open. In contrast, DNA that's methylated, when it gets repackaged after DNA replication, gets packaged in a closed conformation. And methylation seems to work through a lot of different mechanisms. It works through histone modifications. It works through acetylation, through methylation of histones, through ubiquitinization of histones, through methylation of lysine 4, but through additional mechanisms that have something to do with the overall accessibility and higher order structure of the chromatin. All of these things are really unknown and unstudied. Okay, next step. Now we're getting closer to studying about methylation during development. So we did this simple experiment in which we took some housekeeping genes, 2 housekeeping genes, and we asked what is their methylation pattern. Okay? This was before the day of global analysis of the genome. We did it using restriction enzymes. So I believe this is the APRT gene. The APRT gene, this is the 5' end, this is the 3' end. The 5' end looks like this. These are all enzymes that are sensitive to methylation, and MSP, HEP. I think this is HEPA, HAHA, Somebody remind me what this is. I don't remember. This is AVA2, which is also sensitive to methylation. EXA1, which is also sensitive to methylation. And we took these enzymes and we used them to analyze a typical housekeeping gene. Okay. And what we found was, as you can see here, all of these sites in the 5' region of the gene were unmethylated. And this was true in every tissue that we looked at. It was true in sperm, it was true in liver, and spleen, and other tissues as well. Yet this part of the gene looked like it was methylated. So there was something about this part of the gene that was unmethylated, and it was unmethylated constitutively. And this was already an interesting phenomenon, because this is what you expect. If there's a relationship between DNA methylation and activity, a housekeeping gene which is active in every tissue, you would expect it to be unmethylated in every tissue. And indeed, nobody had ever looked at that before. Everybody had looked at tissue-specific genes, and they saw that it's unmethylated here and methylated in other places. Well, it turns out that a housekeeping gene has the pattern that you would expect. It's unmethylated everywhere. It turns out that this, as you can notice from here, that there are a lot of sites here for these enzymes. So these are all enzymes that recognize CGs. Right? And it turns out that all the housekeeping genes have a region like this where there are lots of CGs, and this ultimately became called by Adrian Bird a CPG island. Right? And it turns out that all housekeeping genes have these CPG islands, and CPG islands are basically unmethylated throughout development. So it turns out housekeeping genes have a special structure. Their promoter regions and upstream regions have a special structure that makes them unmethylated in all tissues. Tissue-specific genes, as I mentioned before, are initially methylated, probably during development. They're probably methylated to begin with, and soon we'll see how that happens. Okay? Because they're methylated everywhere. But they're unmethylated in the tissue of expression. So it has to happen something like this. Again, we were guessing. We were just trying to learn about development by looking at different tissues. So this is my son Yoav, for those of you who don't recognize him. And here is a close-up of his cells from his nervous system. And you can see there are housekeeping genes, which are generally unmethylated. There are other genes which are methylated. The genes that are active in neuronal tissue get demethylated specifically in the brain. In muscle cells, there are other genes that are specific for muscles that get demethylated in a muscle. And in the liver, there are other sets of genes that get specifically demethylated in the liver. Somehow this worked. Somehow this is the way it works. We didn't know a lot about how this methylation pattern comes about, but it was easy to see, because there was methylation everywhere, that in terms of development, these genes had to start off as being methylated somewhere, and then there was specific demethylation in tissues of expression. Yoel Yisraeli did this experiment, which was a real eye-opener to us. When we got the results, we really didn't understand it. So I told you about all sorts of experiments where you take a look at fibroblasts, and you put any gene into a fibroblast methylated, it gets turned off. You put it in unmethylated, it's on. Even genes that are not well expressed. Okay? You put them into the cell, so there's a little bit of expression, but if it's methylated, there's less expression. Incidentally, this sort of, we try to understand this in terms of the transfected cell, of the genes in the transfected cell. If you take, for instance, a globin gene and put it into a fibroblast, so if it's unmethylated, it gives a low amount of transcription. If it's methylated, it gets very, very low amount of transcription. Right? So we understood this as meaning, well, if you take a look at the fibroblast, let's look at the endogenous globin gene. What does it look like? So in the fibroblast, it would be methylated. And of course, it's completely shut off. And so we used this as a model. We said, okay, the transfected gene, if it's methylated, gets turned off. That means that in the cell, the endogenous gene is not expressed. Why? Why isn't it expressed? Because it's methylated. If it were unmethylated, in other words, putting in an unmethylated gene, it would have been expressed. And so we imagined that these in vitro experiments, these transfection experiments, were reflecting what's happening in vivo. Surprise came when we took a myoblast cell. You can grow myoblast cells in culture. You can induce them to become muscle. These are sort of precursors of muscle cells. You grow them. You take away the serum, and the cells differentiate into muscle. Real muscle, they pulse, they contract, just like muscle cells in vivo. Right? The actin gene, for instance, is one of the genes that is active in muscle cells. In myoblasts, it's a little bit active, but it's at a low level. When you induce the cell, actin gets induced. So we did a transfection experiment. We took unmethylated actin, and we took methylated actin, and we introduced it into a myoblast. And we assumed we were going to get the same results that we got with a fibroblast. Now this, I'm sure nobody older than or younger than 50 knows what this is. But this is an assay for gene expression in which the gene has a reporter, the cat gene, and then using thin layer chromatography, you can see if the cat was converted, whether it got acetylated. And the assay looks like this. If you get a spot, these are the starting materials for the cat assay. If you get a spot here, it means that you had activity. If you don't get a spot, it means that there was no activity. That's the way the assay works. It's a reporter assay. You're laughing because, right. Danielle did about 10 million of these assays like this. Okay. Okay. So we took the unmethylated actin gene. We put it into cells. As you would expect, it's active, okay, at least at some level. Then we took the methylated actin gene, we put it into the same cells, and it was also active to the same degree. And it took us a little while to figure out what was happening. What was happening? What's happening is that this is a muscle cell, a muscle precursor cell. Okay? Now what happens in vivo? In vivo, let's say the globin gene, what happens in a blood cell? It undergoes demethylation in the blood cell, in a mature blood cell or a precursor blood cell. Right? And that was happening here. In other words, since this is a muscle cell, it knows to recognize actin. It knows that this is actin. And one of the things that happens during normal development is that it recognizes actin and it opens it up. It demethylates it. It changes its chromatin structure. And this is what allows it to be expressed. And so when we put in a methylated gene into a myoblast, that's what it did. It did what it's supposed to do. It demethylated the gene. And then once it demethylated the gene was active. Okay? So then we said, how does it recognize alpha actin? How does it know to demethylate it? Okay? So, of course, in order to know to recognize the gene, you have to recognize a sequence. So we started looking for sequences that were important for demethylation. Are there sequences along the gene that are required to cause demethylation? And in fact, we found something like that. So it's shown here. This is in the full gene. The full promoter has this sequence here. And this is actually part of the enhancer, part of the alpha actin enhancer. So we made constructs that don't have this sequence. Okay? So now what happens? You put in alpha actin, unmethylated, you get activity. It doesn't need this sequence to activity. It doesn't need this sequence for transcription. Right? And here's the methylated gene. But the methylated gene needs this sequence, doesn't have it. It can't demethylate it. And because it can't demethylate it, the gene doesn't get turned on. Okay? So we learned a very important lesson from this experiment. And actually, this is a general concept that is very confusing for people who work on DNA methylation, very confusing for people who work on gene expression. But it's a very, very important principle. And that is that this is all common sense. You look in the organism, you see that the gene is methylated everywhere, and you find one tissue in which a specific gene is demethylated, right? That tells you that this is not caused by demethylation. There has to be something that recognizes the gene. This is a specific phenomenon, gene-specific phenomenon, cell-specific phenomenon. There has to be a factor. There has to be something that recognizes the DNA. And indeed, the demethylation here is really secondary. The acting force here is some sort of factor that recognizes the DNA. It's only present in myoblasts, and it's what brings about both demethylation and chromatin decondensation and the activation of the gene. Has to be factors that do it. This is not a global process, it's a specific process. Okay, this is Zeev Parush did this experiment in which we transfected into cells, into these same myoblast cells, the actin gene, but we transfected it in not as a stable transfectant, but as a transient transfectant. Okay, so these molecules don't get integrated, they don't replicate. And we were able to show that even without replication, these things undergo demethylation. So here's an example. Here's this is all by southern blotting. We introduced the gene by transfection, by transient transfection. This is cut with some restriction enzyme that shows you the gene itself. And then this is cut with a methyl-sensitive enzyme. You can see that some of it is demethylated. This is HEPA2. This is MSP, which cuts it by itself. You can see that it got demethylated. Okay? And of course, all of this is done with a molecule that's labeled at its 5' end. And we're looking at the label. They don't undergo replication. And in any event, it still has the same label, so that molecule didn't undergo replication, and yet you get demethylation. This was not the first indication that demethylation is active rather than passive. It was not the first indication. Somebody did this on the delta crystalline gene years before by inhibiting replication and showing that you get demethylation. But this was the first molecular demonstration that this is indeed the case on molecules that are labeled and do not undergo regulation. Until today, we really don't know how these molecules undergo demethylation. The biochemistry of demethylation, and in particular tissue-specific demethylation, is completely unknown. Okay, so from these studies, which were the beginning of sort of the developmental studies, we drew this picture of the genome. There are regions that have very few CGs. It's underrepresented, but most of them are methylated. There are a lot of tissue-specific genes here. Not all of the tissue-specific genes, but a lot of tissue-specific genes are in this fraction. And housekeeping genes and other genes that are also tissue-specific look like this. They have promoters with CPG islands, and the CPG islands are constitutively unmethylated. But a gene like this can be 2 megabases long. The rest of the gene can be methylated, like here. So there's a bimodal pattern of methylation. Almost everything is methylated, but CPG islands are unmethylated. There are variations in different specific cell types. Right? These tissue-specific genes are sometimes unmethylated in a cell type. But overall, this is the pattern. Everything's methylated, CPG islands are unmethylated. How does this pattern come about? How does the organism develop this pattern of methylation organism-wide? First hint. Okay? So these are experiments that were done on specific gene sequences in the mouse. We assayed methylation. You'll see that we assayed methylation in tissues in which there are very few cells during spermatogenesis or oogenesis, or in oocytes themselves, or in early embryos at the 8-cell or 16-cell stage. Okay? In order to do that, we developed an assay in which basically what you do is you look at the DNA and you cut it with a restriction enzyme. If it's methylated, the restriction enzyme won't cut, so then you can use PCR to amplify that piece of DNA, and you'll get a product. However, if the DNA is unmethylated, the restriction enzyme will cut it. You will no longer be able to make a PCR product. And so this sort of, it's not an accurate assay. It's partially active. In the hands of Tal Kafri, who did these experiments, it became almost quantitative. But you have to realize that it has limitations. Anyhow, so you see the following pattern. First of all, DHFR, which is a housekeeping gene. And if we look now, well, maybe we should start. Well, where should we start? First of all, 6.5-day embryo. This is post-implantation. Post-implantation. This gene is unmethylated. The open boxes indicate unmethylated. It's a CpG island, as we would expect, and it remains unmethylated in the organism, of course. Other genes are methylated in the 6.5-day embryo, consistent with the picture I just showed you, that lots of places are methylated. CpG islands are unmethylated. In this study, we only looked at one CpG island, but it turns out this is true for all the CpG islands, or a lot of the CpG islands. Okay, how does this pattern come about? Okay, so just taking a little peek, you can already see that in the blastocyst, there's no methylation. Everything's empty. Okay? So there has to be de novo methylation here. But let's take a look at the process coming from the beginning, starting with the gametes. Okay? And starting with the gametes means to go back during gametogenesis. Okay, so first thing is, if we look at an early stage of gametogenesis, this is at the stage where we, as the observer, cannot distinguish between what's a male or a female yet. This is at about, it says 12.5 days, probably more, 12 days or 11.5 days. Morphologically, if you isolate these gametic cells, you still can't tell whether it's going to be a male or a female. Everything's unmethylated. Okay? Then if you take a look at the beginning of spermatogenesis or the beginning of oogenesis, again, everything's unmethylated. And then there seems to be a process of de novo methylation. De novo methylation, but the islands remain unmethylated, both during oogenesis and being spermatogenesis. At the end of the process, you get sperm. Sperm looks very much like this. It's methylated in almost all sequences. CpG islands are unmethylated. Oocytes really look like this also, but because of the nature of the genes that we looked at, it turns out that in oocytes, it looks like there are more unmethylated ones here. And this is an exaggeration because of the way we selected the genes that we looked at. Basically, oocytes look the same way. Then what happens? This sperm gets together with this oocyte. You get an early embryo. Look what happens in the early embryo. It all gets erased. Okay, so let's now go through this quickly. We start the embryo. The pattern from the gametes gets erased. We pass implantation. At somewhere between here and here, we get de novo methylation. Islands seem to be protected. The gametes do not come from here. The gametes are formed from tissue that had already become de novo methylated. Okay? And then they undergo demethylation. Just like this demethylation, there's a demethylation in gametic tissue. Okay? And it's demethylated for a while, just like this, and then it undergoes de novo methylation in a process that's very similar to this. So in animals, or at least in the mouse, there's a double cycle. Okay? If we look at the embryo, things get erased, and then the pattern gets reestablished. If we look during gametogenesis, the same thing happens. The pattern gets erased and then gets reestablished during gametogenesis. Like gametogenesis is sort of a separate organism that does the same thing. Okay. So what does this mean? We now know that between here and here, there's a process of de novo methylation. We know that at this time, the de novo methylases, not the maintenance methylase, the de novo methylases, which are called DNMT3A and DNMT3B, are at a very, very high level, and they're presumably able to methylate de novo the entire genome. Okay? Somehow CpG islands are protected from this process and don't get methylated. Okay? Overall, if you now look at the overall picture, this is the way that you establish the pattern that we saw in that diagram. Everything gets methylated. CpG islands are protected. That's the concept. Okay, so Michael Brandeis was interested in understanding how these CpG islands are protected. What protects them? How does it? And of course, CpG islands are protected. It has to have something to do with sequence. In other words, at the time when there's de novo methylation, you can say demethylation is global, so everything gets methylated. But as soon as you say that there are a whole bunch of places that get protected, they have to be recognized. And so it has to be some sort of sequences involved. We thought, and it turned out we were wrong, that the sequence that is recognized is simply a high concentration of CG. Okay? In places where there's a high concentration of CG will be protected somehow. Okay? This is, it was probably a bad assumption to begin with, mainly because it's hard for a human being like us to imagine how you would build a system to recognize concentration of CG over 1,000 nucleotides long. It's a little hard to conceive of something like that, right? And it turns out that it doesn't work like that. So what Michael did was very simple. He actually, Dale Frank started this, and then Michael continued it. He took this APRT gene. This is the CpG island, which is unmethylated. And he started making fragments of DNA which were either in the CpG island or were not in the CpG island, and transfected them into ES cells, or what we used then was a model for embryonic stem cells was F9 teratocarcinoma cells, a very close model. It turns out that these cells are a very, very good model for what happens at about the time of implantation, about the time that you get this de novo methylation. Okay? And the reason I say that is because if you take an unmethylated island and put it into one of these ES cells, so here it is without any digestion, and if you put in an island sequence and ES cells, and then you test with HEPA2 or with HAHA, two enzymes that are methyl sensitive, they remain unmethylated. But if you put in a non-island sequence, they get de novo methylated. So the process of de novo methylation is ongoing in these ES cells. These ES cells dynamically de novo methylate. And at the same time, they know how to protect an island sequence. They don't methylate them. Okay? So I think this was maybe Dale's experiment. He transfected in these. He found that if you transfect in a gene like this into the cell, the transgene doesn't get methylated over here in the CpG island region, but outside the CpG island region it gets methylated, and it's exactly the same pattern as the authentic APRT gene in the cell. Okay. Then Michael continued this. I'm not going to go into this a lot, but you can see how it works. I think here what he did was he started with methylated sequence. It turns out that these cells can also demethylate islands. So if you put in a methylated island, it gets demethylated in a HEPA, digested it. This is the control MSP. This is HAHA, also digested it. But if you put in a methylated non-island from the APRT region, It stays mostly methylated, both in the HEPA and the HAHA sites. CFO is another name for HAHA. So then Michael started dissecting the system. So let's see, this is the island region. This whole thing is now amplified. It's the whole island region. And he said, what in the island region is necessary in order for it to be recognized and protected, or for it to undergo demethylation? Okay? So the whole thing, if you put it in, gets demethylated. Plus here means demethylated. If you take this region, it gets demethylated. You take this region, it gets demethylated. However, if you now go down smaller, take this region over here, it doesn't get demethylated. And here's some of the results. Here's this region over here. You can see HEPA doesn't cut it. Didn't get demethylated. But this sequence right next to it got digested. Did get demethylated. Right? So he did this analysis across the whole CPG island region. And we right away noticed that what seemed to be important were the SP1 sites. SP1 is a factor that binds many CPG islands, many housekeeping gene promoters. If SP1 was present, it seemed to be that protected against de novo methylation or caused de novo methylation. That's why this one works and this one works, but this doesn't work. Right? Okay, and this one doesn't work. Right. Okay. So then he even looked at it small, he did SP1 itself, he made a construct now, that sort of a reporter construct in which you could measure methylation in a region, and then he put in sequences to see if those sequences can induce demethylation of his reporter. Okay? This is the reporter. You put the sequence in over here like this, and right near it there's a HEPA site, there's an EXPA site, there are HAHA sites nearby, and he tested whether this sequence, this small sequence, does the work of protecting against de novo methylation. And SP1 works. You can see it gets demethylated. Without anything, nothing happens. This is just CG, 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 CG. Doesn't look like CG, CG, CG. Looks like G, but it should be CG, 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 CG. It doesn't get unmethylated. That's not good enough. All CGs, not good enough. Doesn't have. So here's SP1. It works. Anyhow, so there are cis acting sequences. SP1 is one of them. Maybe there are other ones. These cis acting sequences work at a distance. So if you're nearby, the site is nearby, you get a lot of demethylation. If the site you're looking at is far from this sequence, you get very little demethylation. It works in a regional manner. Then we took a mouse. We took an APRT gene and we made a mouse, a transgenic mouse, without the SP1 sites. Okay? So first of all, it's important to understand when you make a transgenic mouse, you introduce the DNA basically at the stage of the fertilized oocyte. Okay? So what's going to happen to this DNA? Put it in. It goes through blastula, okay, and then when it comes to the time of de novo methylation, it's going to get de novo methylated, depending on what it is. In other words, transfected DNA is going to undergo the exact same process of endogenous DNA in terms of this de novo methylation. It's a great assay. So just quickly, I don't want to spend time on this. He took the wild-type APRT gene, looked at the promoter region, looked at the CPG island, and you can see it became demethylated. This foreign CPRT housekeeping gene, as you would expect, right? But without the SP1 sites, it remained completely methylated. So again, you need cis-acting sequences. Oh, this doesn't affect the phenotype. It's an external APRT gene. It has its own APRT gene, doesn't care. Okay, this just shows you basically, again, how this works. So this is the reporter sequence. You can put into it what we ultimately ended up calling an island demethylation element. Right? You can put it in here. Without anything, all the reporter sites, SAL1, SMA1, HAHA1 are all methylated. If you put in one of these sequences, they all become demethylated. If you're too far away from the island demethylation element, like this EAG1 site, which is 300 nucleotides away, or the SAC2 site, which is 500 nucleotides away, you don't get demethylation. Works in a regional manner. Typical islands in the genome have a number of these sequences. Okay? Now, it turns out to be a little bit more complicated than that. We're going to get to it. Now, this shows, this is really the ultimate test of what methylation does in vivo. Or at least one of the things that of methylation does in vivo. This experiment was done by you, no? Right? This was done by Zahava. A very actually heroic experiment. And what we did was the following. Okay? You have to follow this a little bit carefully because the genetics are a little bit complicated, not a lot. Okay. For today's generation, it's not complicated. Okay, so we took a normal globin gene, and it had these enhancers, but they're irrelevant to our discussion. And we put in an island element, and we surrounded it with LOX-P sites so that it could be removed by Cree. Okay? So then we took this mouse. Right? So again, this mouse has an island element with lock sites, right? And it's next to the globin gene. Okay, this is the normal human globin gene. Now, we cross this animal with an animal, a mouse that expresses Cree from very, very early development, from the first stages of development. It actually even expresses it already in the oocyte. Very, very early expresses Cree. What happens? Right away, this island element gets removed. So now we have our construct, but without the island element. What's going to happen? It's not an island, this whole thing. It's going to get de novo methylated. And indeed, it gets de novo methylated. Okay? Post-implantation, you look at it, it's de novo methylated. And it'll be de novo methylated throughout the organism. Why will it be de novo methylated throughout the organism? Because once it's established at the time of implantation as being methylated, the pattern then gets maintained. Okay. Alternatively, we took the same mouse and we crossed it with an animal that has an inducible Cree. But we didn't induce it yet. So at the time of pre-implantation, Cree isn't working yet. The island element remains in. Because the island element is there, this gene does not get de novo methylated. It gets protected. So it's not methylated. So now we have two mice that are basically identical, exact same integration site. Integration site is very important when you're dealing with transgenic mice. Okay? Same exact integration site, but this one's methylated, this one's unmethylated. Unfortunately, they're not identical because this one still has the IE. But that you can get rid of. Of course, you can take the adult and induce Cree and remove it. And then you really have two animals that are identical to each other, except that at the time of implantation, one got methylated, the other didn't. Okay? This experiment now allows us to ask, what is the role of methylation in vivo? This experiment had to wait until we were able to manipulate DNA methylation in vivo. To manipulate DNA methylation in vivo, you have to know the rules of how it gets methylated or not methylated. So this experiment essentially had to wait until we knew something about development. What happened? Okay, so what happens? Do we have data for this, or should I just tell you about it? So if you look at so here's an example of our globin transgene. Okay? In the liver, unmethylated, there's activity. It's not a lot of activity, by the way. It's exaggerated because we did PCR. But it's not a lot of activity because in the liver, globin is not really active. Right? When it's methylated, it had about 40 to 50-fold less. Spleen, also inhibited, a little less. Brain, inhibited. Heart, muscle, inhibited. Kidney, inhibited. So what is methylation doing? Basically, methylation, again, I have to point out that we're not looking at any tissue-specific activity. We're looking in all of these experiments at what happens to the globin gene in tissues in which the gene is not expressed. Okay? When it gets expressed, methylation is not relevant. Because in order to get expressed in the tissue of expression, there's a mechanism for removing the methylation. So if methylation plays a role in this gene regulation, it has to be in tissues in which the gene is not expressed. And so what it looks like is this methylation is providing a mechanism for keeping the level of globin transcription at a very low level. And we now know from other experiments that are similar to this, that were done by Alon Goren, that methylation by itself lowers the amount of Beigel transcription by something like 20-fold, 30-fold, something like that. There are more mechanisms for inhibiting globin expression. Methylation is not the only one. Right? The difference in globin between an expressing cell and an not expressing cell is gigantic. It could be 8 orders of magnitude difference. And methylation is only providing for part of that. Right? So there are more mechanisms. A lot of it has to do with the tissue-specific factors that are used in the tissue of expression. Right? So methylation plays a role in keeping down the level of gene expression, of course, in non-expressing tissues. So it seems to have, and this is actually a nice concept because methylation is occurring globally. Okay? It's occurring globally without having to recognize any of these genes specifically. We're sort of answering one of the questions that we started with at the beginning. It doesn't work like bacteria. Bacteria, you want to repress a gene, you have to recognize it, bring a repressor that recognizes that sequence, and turn off the gene. Here, many tissue-specific genes are kept at a very, very low level of expression throughout the organism simply because they're part of this global mechanism of just plain methylating everything. Right, so global mechanism for keeping down gene expression. This is probably one of the most significant mechanisms of DNA methylation. We'll talk a little bit more about that later. Okay. Tamar Hashim-Shoni went on to show that in these mice, the structure of the globin gene is different, whether it's unmethylated or methylated. This indicates that demethylation controls protein structure in vivo. Okay? So here, if the gene's unmethylated, we found that H4 was acetylated, H3 is acetylated at this site, lysine 4 is methylated at this site. This automatically happens if the gene is unmethylated. And within every tissue we saw this. Okay? On the other hand, if the gene was of the methylated flavor, okay, was not acetylated, didn't have methylation at lysine 4, and was methylated at lysine 9. So all of these things here automatically happen as a result of the DNA methylation. Okay? One of the things, we don't know much about this, how this happens. Okay? One of the things we know, we think we know, I'm not 100% sure it's true, that deacetylation as a result of methylation, when the DNA is methylated, there's a factor, MECP2, that can bind and recognize these methyl sites. And MECP2 is known to recruit a deacetylase. So it could be that methylation, presence of methylation itself, brings about this deacetylation of histones by some sort of mechanism like this. There are probably other mechanisms as well. In fact, the latest issue of Nature has a paper by Adrian Bird on a mechanism for controlling lysine 4 methylation, which is a little bit different than this. Okay? So this could be one of the mechanisms. So again, this pattern is established at the time of implantation. Then it's maintained by the mechanism we talked about, maintenance methylase. So it appears in every tissue. No longer, in these somatic cells, you no longer have the machinery for de novo methylation. You no longer have the machinery for protecting CpG islands, but the pattern is maintained by this mechanism. And, okay, so the bottom line is, I'm going much, much slower than I anticipated. So, this rate, we're not going to get very far, actually. And so, the overall picture is what's so far. What's important? Why is methylation important? At the time of implantation, you set up this pattern. In my opinion, this is the only time in the organism that methylation per se works as a mechanism. Every other place that you see differences in methylation, whether it be demethylation in a tissue-specific form or de novo methylation in a developmental or tissue-specific form, all of those are directed by specific tractors that have to recognize sequence. There, the methylation is secondary. You need it. And I'm going to show you later why you need it. You need the methylation. Methylation plays a role. But it's a secondary mechanism. The one place in the history of the organism, in the ontology of the organism, that methylation per se plays a role is at the time of implantation. And even there you can argue with me because there's a mechanism for recognizing the islands. Right? So what gets methylated is also controlled by sequence. It's true. Very, very true. Right? It all has, of course, everything in the end has to be written in the sequence because that's what we get from our parents. Right? But that stage of implantation is the only stage that's really controlled by methylation. It works like a global mechanism. It methylates everything. And more than that, it provides a mechanism for maintaining chromatin structure later on. Every time the cell divides, it disrupts the chromatin structure. You have to reconstruct it. DNA methylation is there because it gets maintained. And it plays a very, very big role in aiding, facilitating this building again after replication of this is really the bottom line of epigenetic. You hear a lot of definitions of epigenetics. This is the bottom line of what's epigenetic. Right? Being able to maintain long-term chromatin structure in the organism. Right? And we're going to define this a little better in the next session. We're never going to get to replication timing. We're never going to get to all those other things, but at least we'll all know about DNA methylation. Okay, thank you.